So today we're going to go over the answers to the review that I gave you on Monday. And then you do have another test review on protein synthesis and mutations on Schoology. That's your actual assignment today. And then go ahead and prepare for your FRQ as well to make sure you're ready for tomorrow's test. So let's go through our test review. So the first one is to compare and contrast RNA and DNA. Well, you can't see what I'm seeing. Okay, so RNA could be single-stranded. It's just one strand. DNA would be double. That's why we call it a double helix. Single, double-stranded. RNA and DNA are both nucleic acids, which are one of the 4 major biomolecules. They both have the monomers of the same. Those are going to be nucleotides. And the nucleotides are all going to be composed of the same 3 things. Well, not the same, but parts, same parts. A phosphate, a sugar, and a nitrogen base. However, the sugars are going to be different for an RNA and a DNA molecule. For RNA, the sugar is going to be a ribose. For DNA, the sugar is going to be a deoxyribose. And that's kind of how they get their names. Ribose, RNA, deoxyribose, DNA. The nitrogen bases they're going to have are going to be your guanine, cytosine, adenine, but they're going to have a different 4th base. These are base, sorry. For RNA, the 4th base is going to be uracil. For DNA, the 4th base is going to be thymine. And the last thing that they have that's different is that DNA only stays in the nucleus. RNA can leave the nucleus. It's small enough, it stays in the nucleus and in the cytoplasm. It can move around wherever it wants. So we're going to complete the chart below, and then we're going to compare them to our amino acids. So the first one we have is mRNA, are the code with the codons, and that's going to be in both the nucleus and the cytoplasm. Remember, it can move. mRNA is also involved with both transcription and translation. We use it first in transcription to copy DNA, and then we use it in translation to make the proteins. Then we have our tRNA and our amino acids. Those only play a role in translation because they're only occurring in the cytoplasm. So tRNA and amino acids only in the cytoplasm, only with translation. mRNA is both in the nucleus and the cytoplasm, and it's both transcription and translation. So when you're making your mRNA, it's the complementary strand to DNA, except that it doesn't have any thymines. It only has uracil. So T still goes with A, but this time A goes with U, C goes with G. GGC would be CCG, CTA would be GAU, then we have GUU, then we have UGA. The tRNA, that's just the anticodons for the codon. Those don't code for your amino acids. They're just there to register the mRNA strand. Again, tRNAs don't code for amino acids. So tRNA would kind of sort of be the same as the DNA template, except it doesn't have T's, it has U's. So I'm just going to copy this, and every time I see a T, I'm going to put a U. So UAC, GGC, CUA, CAA, and ACU. But again, I'm not using tRNA to code for my amino acids. I'm still going to use my mRNA. So AUG, I have A, then I have U, and I have G. That's my start. R my methylene, whichever one you want to write down. And then you just go from there. You have your proline next, your aspartate next, then your valine next, and then finally you have a stop. So just keep in mind that amino acids go to the mRNA. Any questions on this first page? All right, going on to the back, this process right here, we have a cell, then we have a nucleus, and then the cytoplasm. So this is protein synthesis. In the diagram, what are the steps? We have step 1 right here, and we have step 2 right here. Step 1 is going to be transcription. And step 2 is translation. We're going to describe the process. For transcription, what happens is that the DNA is copied into an mRNA strand. For translation, the process is the mRNA is coded into a protein. Number 5, in the diagram above, list and describe the function of all 3 types of RNA. So we have A, B, and C. A is right here. That's mRNA. B is right here. That's my tRNA. And C is right here. That is my rRNA. Describing the functions, mRNA is responsible for copying the DNA. Then it's going to take that info into the rRNA. tRNA, the function of that one is to carry the amino acids to the rRNA. So that way it can make the proteins. And then finally, rRNA, that's the ribosome. That's where all the translation happens. It reads the mRNA, it brings in the tRNA, and then it's making proteins with those amino acids. Number 6, in the diagram above, where do the steps of protein synthesis occur? List the letter and state the location of each. So where do they occur? Transcription is going to occur in E, the nucleus. Translation is going to occur in F, the cytoplasm. And then there was a typo on here. I'm sorry, I just noticed that. These are actually supposed to say letter D. Sorry. So on number 7 in the diagram above, what does the letter D represent? Well, D is right here. And D is DNA. In eukaryotic cells, it's in the nucleus. In prokaryotic cells, it's in the cytoplasm. Because remember, if you're a euc, you have a nuke, and pro means no nucleus. Any questions on this page, 3 through 7? All right, let's keep going. Number 8 in the diagram above, a chain of G. G is this letter right here. That's an amino acid. Is going to be linked together with peptides, peptide bonds, or polypeptides, whichever one you wrote, that's fine. And it's going to form the letter H, which is this letter right there. H is a protein. Number 9, in the diagram above, what does letter I represent? I is right here. And it's circling 3 bases. So that's going to be my codon. Which type of RNA has them? That's my mRNA. Number 10, what does the letter J represent? J is right here. J is circling the 3 bases at the bottom of this tRNA. So that's my anti-codon. Which type of RNA has them? Well, I just said it, tRNA. Number 11, what organelle is a protein factory? That's a ribosome, because that's where we're actually making the proteins in this process, in the ribosome. The ribosome is reading mRNA, it's getting the amino acids, and it's making a string of proteins. Number 12, what process is happening in the image below? How can you tell? Well, if I'm looking at this image, I can see 2 strands right here, and then I can see 1 strand right here. So if I have 2 strands, that has to be DNA, 1 strand, that has to be mRNA. This has to be transcription. And I know it's transcription because, again, I'm seeing my DNA and I'm seeing it being copied into an mRNA strand. DNA has T's, mRNA has U's. Number 13, label the letters in the image above. So some of these were kind of confusing. Hopefully you got them. A is pointing to this enzyme right here, and that is an RNA polymerase. B is pointing to this base right here, the letter A, and that's a free RNA nucleotide. Or it's just a free nucleotide. I'm only saying RNA because it's going to make an mRNA strand. You have C pointing to this bottom strand, which this one is the DNA coding strand. So C is going to be the DNA template strand. And that's because it's being the template for what we're copying. And finally, D is pointing to mRNA. All right, any questions on 8 through 13? Any? All right, let's keep going. Number 14, which process during protein synthesis is taking place in the image below? How can you tell? Well, here I see a lot of things going on. I can tell this is my mRNA because it's a single strand. I can tell this is a tRNA because it has only 3 bases and it has an amino acid. So I could only assume that this is translation. Now I'm going to label the letters in the image to the right. My mRNA, it has to be my single strand, so that's E. tRNA is this funky shape right here. My amino acids, that's going to be the little circular ball, so that's D. My protein, my polypeptide chain, that's my amino acids together, so that's B. An anticodon is on my tRNA, so that's going to be A right here. My codon is a set of 3 letters on my mRNA strand, so that's going to be my G. And finally, my ribosome is this funky globular shape right here. That's F. Any questions on translation? All right. 16 starts off with mutations. So what is a mutation? It's just a change in DNA. Number 17, are all mutations bad? No, they don't all have to be bad. Remember, that's how organisms evolve. That's how they get different traits. And if those traits are beneficial, if they help them to survive, then that's a good mutation. We want to keep those. Number 18, when might a mutation be considered neutral or have no effect? Whenever that change is going to code for the same protein. When it codes for same protein. So if you change a base by substitution and it still gives you the same amino acid, you're still going to get the same protein. Let me give you an example. If you have a strand Let's take proline, for example. If you have a strand that says CCA, and that last A gets changed into a G, well, it's still going to code for protein or proline. You won't feel a difference. So that's CCA and CCG. You're still going to have the same effect. So if you change that with substitution, you're still going to have the same amino acid, still going to have the same protein. Nothing's going to change. Number 19, when could a mutation be beneficial? Just like we said here, are all mutations bad? No. It could be beneficial when it helps the organism survive. When it helps the organism to survive, then it's a beneficial mutation. Then we want to keep it. That's how organisms can evolve. Any questions on 14 through 19? All right. Last page. Identify the types of mutations below. Here we have time to dream. Then we have the I changing to an A, so it says tame to dream. That is substitution. All we did was change a letter. Now we're taking out the I and the M. So instead of time to dream, it's Tito dream. That is deletion, which is a type of frame shift mutation. Time to dream, we're adding in that extra I, so it's team et o dream. That is addition. And that's another frame shift mutation. Number 21, identify the types of mutations. Here we have met-lis-phi-gly-stop. We're taking out that extra U. That's leaving, the picture's kind of hard to see here, but there's an arrow taking out the U. So that's deletion. And notice how it doesn't stop and it codes for completely different amino acids. Then on B, again, I don't know if you can see that arrow, but it's pointing in. It's adding in that extra U, and it's causing it to read UAA, which is stop. So this is addition. And just because it added that one extra letter, it's stopping that protein from being made. So instead of having a strand of amino acids, you just have one. So that protein is not valid. It's not useful. We can't use it. Number 22, mutated DNA in somatic body cells occur frequently. Why would this modified mutated DNA not be passed along to the offspring or children? Because it's in your body. It's not happening in your sperm or egg cells and your gametes. It's happening in your body cells. So it stays in your body. And finally, 23, how do DNA instructions get expressed as genes or traits? With proteins. We turn them on with gene expression, which is what we talked about yesterday. So those proteins carry the traits and they get turned on with gene expression. So the mRNA gets to make them when it wants to. If it's turned off, it won't. If it's turned on, it will. All right, so that's all I needed to talk about. Go ahead and do your review that's in your folder. And then study, prepare, turn in all of your missing assignments. I did send an email yesterday to everyone that if you are failing, I'll be emailing and calling home tomorrow. So make sure you get everything turned in. If you have any questions about anything, stay on. If not, you're free to go.